Hello everyone, this is for a B.Sc program, subject chemistry. In that, paper 8 biochemistry for 6th semester, session number 33 under DCE-Digital learning scheme. Okay, the topics covered in this session are Krupp cycle and energetics of Krupp cycle. I am Dr. Veena K from Maharani's Science College for Women. Now, so in the previous sessions, I have told you regarding EMP pathway, that is glycolysis, how this glucose is converted into pyruvate. Okay? Once you have got a pyruvate, now three things can happen to pyruvate. In that, one of the processes, pyruvate can be converted into acetyl coenzyme A. Now, this acetyl coenzyme A is required to start the Krupp cycle now. This Krupp cycle takes place in the presence of oxygen. So, it's an aerobic pathway to provide energy. See, most of our energy comes from this Krupp cycle. So, this is a very important cycle because, and this takes place, all the reactions of this Krupp cycle takes place in the presence of oxygen. And this was discovered by Hans Krupp in 1953, for which he was awarded a Nobel Prize. And where does this take place? This takes place in the mitochondrial matrix of all aerobically respiring cells. It is also known as citric acid cycle because the first compound which is stable formed is a citric acid. Since it is having 3 COOH groups, it is also known as tricarboxylic acid cycle. You have learnt in your previous classes, if there is only one COOH group, monocarboxylic acid, 2 COOH group, dicarboxylic acid. Similarly, if you have a 3 COOH groups, it is known as a tricarboxylic acid. Since this citric acid contains 3 COOH groups and this is a starting compound formed in the first step, this cycle is also known as TCA cycle. So, which are the three names for this? Krupp cycle, citric acid cycle and tricarboxylic acid cycle. And remember, it is an aerobic pathway which takes place in the mitochondrial matrix of all aerobically respiring cells. Now, come on, we will go to study this Krupp cycle. See, this takes place in the mitochondria, okay? And you have already learned this in your previous sessions, right? Now, see how the reaction starts. A simple diagrammatic way I have represented here. So, this reaction starts when acetyl coenzyme A condenses with oxaloacetate to form a citrate. See, then citrate will give you isocitrate. Similarly, there are 8 reactions in this cycle. Okay, so this is a citric acid cycle or Krupp cycle. Now, see, this is your pyruvate. How did you get this pyruvate? Through glycolysis. You know, glycolysis is an anaerobic pathway. From glycolysis, 2 molecules of pyruvate are formed from glucose. When this pyruvate undergoes that is pyruvate can be converted into this acetyl coenzyme A, which is a very important step. In the presence of pyruvate dehydrogenase complex. And please remember here, whatever may be the different type of food we are going to take, it may be a carbohydrate, it may be oil or fat, or it may be a protein. All these in the second stage of catabolism has to be converted into this compound acetyl coenzyme A. In whatever the route, it has to be converted into what? Acetyl coenzyme A. And this acetyl coenzyme A enters into this TCA cycle or citric acid cycle. So, you get so many intermediates in this step and they will be the precursor for the remaining other reactions. So, whatever the products come here in the citric acid cycle. So, now let us start studying citric acid cycle. So, remember, from glycolysis what you have got? Pyruvate. From pyruvate what you have got? Acetyl coenzyme A. See, this acetyl coenzyme A condenses with oxaloacetate. Condenses with what? Oxaloacetate in presence of citrate synthetase. In presence of citrate synthetase to form citrate. to form citrate. See, if you write here HHH, it's a tricarboxylic acid. Since the starting stable compound formed in this TCA cycle is a tri containing 3 COOH groups, this cycle is also known as tricarboxylic acid cycle. Since the first compound formed is a citric acid, this is also known as citric acid cycle, named after in the honor of Hans Krupp as the Krupp's cycle. Now, what did you get in the first step? This acetyl coenzyme A condenses with oxaloacetate to form citrate. Okay. See, there are 8 enzymes involved here. So you simply cannot write this citric acid cycle just in words. You should know what are the different conversions taking place in each 8 steps, because as a student of chemistry I have been telling you, you should know how to write the reactions. So very slowly I am going to explain you step by step what happens in each step and how to remember writing the reactions. Okay. So what is formed in the first step? That is citrate. Citrate undergoes isomerization to form isocitrate. Okay. And this is hydration and dehydration step. It forms isocitrate. From isocitrate, what you get? Alpha ketoglutarate. I am going to explain you in all the steps just to make you familiar with the names. Isocitrate to alpha ketoglutarate. Alpha ketoglutarate in the next step converts it into succinyl coenzyme A. Succinyl coenzyme A next step converts into succinate. Then succinate to fumarate. Fumarate to malate. Malate to again oxaloacetate. So why I am stressing this cycle is, again you got this oxaloacetate, and this oxaloacetate again combines with acetyl coenzyme A, where the cycle continues. So the 2 carbons of acetyl coenzyme A will come out in the form of carbon dioxide. Okay, so you have got oxidation reduction reactions taking place in this reaction, that is oxidative decarboxylation reaction taking place in this reaction. So how do you start the study of citric acid cycle? From glycolysis, what you have got? Pyruvate. Pyruvate through oxidative decarboxylation, what you have got? Acetyl coenzyme A. So acetyl coenzyme A condenses with oxaloacetate to form citrate. Citrate has got how many COOH groups? 3. Therefore, it is known as a tricarboxylic acid cycle. So, shall we start studying the reactions now? Yes. See, citric acid cycle takes place in the mitochondria of eukaryotes. All the reactions occur in the mitochondrial matrix. Okay. And this is an aerobic pathway, means most of our energy what we get is through the TCA cycle. Okay. So this enzyme complex, which is same, which participate in the ETC chain, is embedded in the inner mitochondrial membrane in eukaryotes, that is succinate dehydrogenase. So remember, citric acid cycle takes place in the mitochondria of eukaryotes. So as of now, you know, from glycolysis what you have got? Pyruvate. From pyruvate what you have got? Acetyl coenzyme A. Acetyl coenzyme A condenses with what? Oxaloacetate to form what? Citrate. So that the citric acid cycle starts now. See, the first step is the synthesis of citrate from acetyl CoA and oxaloacetate. See, this is your acetyl coenzyme A and this is your oxaloacetate. Okay. Now, so the Krupp cycle begins with the formation of citrate. How many carbon atoms it has got? 6 carbon atoms. Okay. So formation of citrate is the first compound. Okay. Now, acetyl coenzyme A condenses with what? Oxaloacetate in presence of what? Citrate synthetase. In presence of citrate synthetase to form citrate. So since it has got 3 COOH groups, 1, 2 and then 3, it is known as a citric acid cycle or tricarboxylic acid cycle. So this is a reactive thiol group. So practice writing the reaction. This is acetyl CoA condenses with what? Oxaloacetate OAA. What is the enzyme? Citrate synthetase. What is the reaction? Condensation. What is the product you got? Citrate. Okay. Now, for every step you should know the enzyme, because in examination you get a question like this. Write all the 8 reactions they'll not ask you. They'll ask you like this. Write the reaction of TCA cycle catalyzed by so and so enzyme. Suppose if they ask you, write the reaction of TCA cycle catalyzed by citrate synthetase. Catalyzed by citrate synthetase. What you are going to write? You are going to write acetyl-CoA condenses with oxaloacetate. What you get is citrate. Okay. So practice writing the reactions. Now, coming to the next step. So you have citrate now. Okay. This is converted into isocitrate. This is converted into isocitrate. So actually this reaction takes place in 2 steps. Okay. With the citrate first gets dehydrated to cis-aconitase, and this on rehydration gives isocitrate, and the enzyme requires Fe2 plus as a cofactor. So we've got citrate in the presence of aconitase Fe3 plus. This is isomerization reaction which gets isomerized to isocitrate. So now you know 2 steps. Step 1, formation of citrate. Step 2, citrate to isocitrate. Now, in the next step, okay, this is isocitrate. Okay. See, there are 4 oxidation reduction reactions in TCA cycle. This is the first oxidation reaction occurring. See, the carboxyl group on the central carbon C3 is removed as CO2. So, what do you call it as? Decarboxylation. And the OH group on carbon 2 is oxidized to keto. See, this is removed. This group is oxidized to keto group. You know, secondary OH upon oxidation, you get a keto group. Okay. And a molecule, when this is oxidized, you know, NAD plus has to get reduced to NADH. Remember, in any reaction, if one component gets oxidized, the other gets reduced. Okay. Now, that's why you call it as oxidation reduction reaction. So, what is happening in this reaction? This CO2 is getting removed. This secondary OH, the OH group here, getting oxidized to CO group. So, a molecule of NADH is produced, which is an enzyme isocitrate dehydrogenase. Now, how do you read this? See, this is alpha ketoglutarate or 2-ketoglutarate. See, the carbon atom next to this COA functional group is known as alpha, beta, gamma, so on. At the alpha carbon, you have a keto group. Therefore, it is known as alpha ketoglutarate, 2 kg or alpha kg, alpha ketoglutarate. Remember here, a molecule of NADH is produced, because at the final I am going to ask you how many NADH is produced and how to calculate the number of ATP released. Now, we will go to step 4 now. See, in the 4th step, this alpha ketoglutarate is converted into succinyl-CoA. Alpha ketoglutarate is converted into what? Succinyl-CoA. See, this is your alpha Kg, which we got in the previous step. In the presence of NAD plus and coenzyme A, see here, you have this enzyme complex which is very similar to pyruvate dehydrogenase complex and requires the cofactors TPP, lipoic acid, coenzyme A, FAD and then NAD plus. Okay. See, the product produced is succinyl-CoA. What is the product produced? Succinyl-CoA. See, this is the second oxidation reduction reaction in TCA cycle, and in the previous, I told you there is one molecule of NADH produced. See, totally there will be liberation of 3 NADH. So, in this step, the second NADH is produced. See, you can go through here. Alpha ketoglutarate, which contains C5 means which contains carbon, 5 carbon atoms, is oxidized to succinate. See, one carbon is lost as CO2, okay, which is an enzyme alpha ketoglutarate dehydrogenase complex. And this is the second oxidation reduction reaction in TCA cycle. And out of 3, this is reaction which produces the second NADH. Okay. So, the carboxyl group is lost as CO2 and is replaced by what? Coenzyme A. Replaced by coenzyme A. So, see, this is lost. Replaced by what? Coenzyme A. What happens to NAD plus? NAD plus getting reduced to NADH and H plus. A second NADH molecule is produced in the TCA cycle. Okay. So, what is happening in the step 4? That is alpha ketoglutarate is converted into succinyl-CoA. Now, come to step 5. See, this succinyl-CoA, this bond gets cleaved here. It's a very important reaction. This is a very important reaction. Succinyl-CoA is converted into succinate. Succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthetase. Okay. See, when this bond is broken, lot of energy is released. So, the energy of this energy-rich succinyl-CoA is conserved by substrate level phosphorylation, because here, look here in this reaction, whatever the energy released in the bond breaking of this is given to GDP. Okay, that is, this GDP is converted to GTP. GTP is converted to GTP. So, this is the only one substrate level phosphorylation taking place in the Krupp cycle. Again, this GTP gives this phosphate group to ADP. In turn, ADP produces ATP. So, remember, this is the only one step in Krupp cycle. That is, step 5 is the only one step in Krupp cycle where a molecule of ATP is produced. I repeat again. Succinyl-CoA, during the bond breakage of this bond, lot of energy is released. Okay, and that energy released is used in the phosphorylation of GDP to GTP. Now, this is the example for substrate level phosphorylation. Only one substrate level phosphorylation in Krupp cycle, and this GTP can transfer its one phosphate group to ADP and in turn producing ATP. So, this is when conversion of succinyl-CoA to succinate takes place, a molecule of ATP is produced. This reaction is an example for substrate level phosphorylation in Krupp cycle. So, here you get for 2 marks a question like this. It may be like this. Well, how do you convert succinyl-CoA to succinate? Or, which is the only one substrate level phosphorylation taking place in Krupp cycle? Or, write the reaction of Krupp cycle where a molecule of GTP is produced. Okay, so try to write these reactions. Okay, I'm stressing many times because all of you will write in words. You never practice writing through reactions like this. So, practice writing the reaction succinyl-CoA converted to what? Succinate. The energy liberated during the breakage of this bond is given to what? GTP. Phosphorylation takes place to form what? GTP. In turn, GTP can transfer one phosphate group to what? ADP to produce ATP. So, this is the one substrate level phosphorylation taking place in the Krupp cycle. Now, come to step 6. So, you have succinate. This succinate gets oxidized to fumarate. Succinate gets oxidized to fumarate. You know very well, oxidation is addition of oxygen or removal of hydrogen. You take out one H from here, one H from here. Give it to FAD, and what you get is FADH2. So, this is a reaction where a molecule of FADH2 is produced in Krupp cycle. So, this is catalyzed by the enzyme what? Succinate dehydrogenase. This also many times I will ask you. Write the reaction of Krupp cycle catalyzed by succinate dehydrogenase. So, I want all of you to remember all the 8 enzymes involved in TCA cycle, because many times you get a question upon enzyme, what is the reaction taking place. So, what is happening in step 6? Oxidation of succinate to fumarate. What is produced? A molecule of FADH2 is produced. What is the enzyme? Succinate dehydrogenase. Okay, come to next step. See, in the last step you have got fumarate. Okay, fumarate undergoes hydration in the presence of an enzyme fumarase to give you malate, to give you L-malate. See, this is a double bond. You know very well characteristic reactions of this unsaturated compounds or compounds having double bond is addition reaction. Open up this double bond. And when you open up this double bond, add H and OH here. H and then OH, what you get is malate. So, fumarate hydrates, fumarase, that is the fumarase enzyme, hydrates fumarate to L-malate is the step 7 of the Krupp cycle. Now, in the next step, See, in this reaction, what is the enzyme? Malate dehydrogenase. You see, this malate dehydrogenases will complete the Krupp cycle. Okay, and it is the up to here we have learned 3 oxidation reduction reaction. See, this is a 4th oxidation reduction reaction, and the last NADH. I have been telling you in Krupp cycle there are 3 NADH molecules are released. In this cycle, the 3rd NADH molecule is obtained. The 3rd NADH molecule is obtained. So you have got L-malate. This is a OH group, secondary OH. Upon oxidation, what you get? Converts to a keto group. And what you got? Oxaloacetate. And once the oxidation is taking place, what should happen to NAD+? NAD+ should get reduced to NADH and H+. So now, in step 8, we have learned oxidation of malate to oxaloacetate, and this malate dehydrogenase completes the cycle and gives oxaloacetate back. And once you have got oxaloacetate, what should happen? Again the cycle should start. Again oxaloacetate condenses with acetyl-CoA to form a citrate, complete a Krupp cycle. All in all, what are all released? 3 molecules of NADH, 1 molecule of FADH2 and 1 ATP released during Krupp cycle. So, Krupp cycle is a process which takes place in the presence of oxygen in the mitochondria, where the pyruvate obtained from glycolysis giving acetyl-CoA, acetyl-CoA condensing with oxaloacetate to form citrate, and the cycle starts. So just now I have told you all the steps, okay, and also all the enzymes involved in the Krupp cycle. Come on, now let us understand the energetics of Krupp cycle. So, what are released in Krupp cycle? NADH and H+, so I'll write NADH2, FADH2, and then from this GTP a molecule of ATP is produced. Okay. So, how many NADH2 produced? 3 per cycle. How many FADH2 produced? 1. How many GTP in turn will give ATP? 1. Okay. Now, see, from glycolysis, you have got 2 molecules of pyruvate. So, 2 molecules of pyruvate should give you 2 molecules of acetyl-CoA. So, you have to calculate this for, all this is for 1 molecule of acetyl-CoA entering into Krupp cycle. But, I am taking 2 cycles into consideration because you have got 2 acetyl-CoA from 2 molecules of pyruvate. So, what will be the number of ATP molecules produced? You know, 1 NADH is equal to, produces 3 ATP. Okay. So, from this Krupp cycle, 2 molecules of pyruvate, the number of ATP produced is 18. The number of ATP produced is 18. Okay. Now, that is, you can calculate like this. 3 NADH produced. Okay. 3 NADH, 1 NADH is equal to how many ATP? 3 ATP. So, 3 into 3 is equal to 9. For 1 acetyl-CoA. For 2 acetyl-CoA, 9 into 2 is equal to 18, to be more simple. Okay. For 1 FADH2, 2 ATPs are produced. For 1 FADH2, 2 ATP. 1 NADH, 3 ATP. Now, this is calculation for 1 acetyl-CoA. And I have been telling you there are 2 acetyl-CoA because glucose has given you 2 pyruvates. So, if 1 NADH will give you 2 ATP, for 2 FADH2, how many ATPs you should get? 4. Now, this GTP will give you 1 ATP, I told you, from the substrate level phosphorylation in step 5 of Krupp cycle. So, if this applies to 2 acetyl-CoA molecule, number of ATP produced is 2. So, remember, when 2 acetyl-CoA molecule enters into Krupp cycle, when 2 acetyl-CoA molecule enters into Krupp cycle, the number of total ATP released is 24. For 1 acetyl-CoA, 12. For 2 acetyl-CoA entering into Krupp cycle, the number of ATP released is 24. Now, so this completes the carbohydrate metabolism. Carbohydrate metabolism means 2 things you should learn. One is glycolysis. Number 2, what happens to pyruvate, that is the fates of pyruvate. Next, from pyruvate entering into Krupp cycle. So now, let us calculate when a molecule of glucose undergoes complete degradation, means you have to include glycolysis as well as Krupp cycle, the number of ATP released. They'll ask you for 4 marks. Okay. Now, see the aerobic pathway I have explained you in the previous sessions. Glycolysis will give you 8 ATP. When conversion of pyruvate to acetyl-CoA with where NADH is released, which that enters into mitochondrial, that is electron transport chain, 6 ATPs are released, and from Krupp cycle 24 ATPs are released. Okay, so in the aerobic pathway, the number of total ATP released is 38 ATP per molecule of glucose. In an anaerobic pathway, glycolysis 8 ATP, fermentation that is minus 6. Okay, now where the a number of ATP not released, utilized, you have to minus it. So the number of total ATP released is 2. Oh, so they'll ask you for 2 marks, sometimes 2, sometimes 4. Write the total number of ATP released by the complete degradation of glucose molecule. So you have to write both. How the number of ATP is released through glycolysis, number of ATP released from Krupp cycle. So now, all in all, what you have learned through in this carbohydrate metabolism is glycolysis takes place in 2 phases, preparatory phase and payoff phase. Consists of 10 steps, 5 steps in preparatory phase, 5 steps in the payoff phase. From glucose you have got pyruvate. Pyruvate is converted into acetyl coenzyme A. This acetyl CoA enters into Krupp cycle. Ultimately, what are the number of ATP released and the total number of ATP released from carbohydrate metabolism. My sincere suggestion to you all, my dear students, everything you will write in words. That's what I have observed. But again and again I am telling you, practice writing the reactions. First write the heading. What is the conversion taking place? Then write the reactant. Then write the product. Unless you practice writing the reactions, you cannot write any reactions in biochemistry cycles like this. And also, remember the 10 enzymes that are participating in glycolysis, make a list. And the 8 enzymes that are involved in the Krupp cycle, make a list. But because the questions will be asked like that only, name the reaction that is catalyzed by so and so enzyme. Okay, so or how one is converted to another with the help of this enzyme. Okay, so practice writing all the 10 reactions of glycolysis and 8 reactions of Krupp cycle. Okay, thank you.